Right, so are you able to see the screen though?
Yes, ma'am.
All right, okay, dear, switch on your video. Let's start today's
Yes.
class.
Mhm.
So we have to start with transcription today, right?
Yeah.
So see, transcription is the process that, not she, transcription is the process that takes place in both prokaryotes as well as eukaryotic organisms, right? So basically, transcription is that process wherein the DNA, from the DNA, the RNA will be formed, or particularly you can say the mRNA will be formed. Now, in the case of prokaryotic organism, it takes place in the cytoplasm of the cell. And see, since this is a very, you can say, complex procedure, so it involves the certain enzymes that are associated with it. Okay, like transcription cannot take place without the transcripting enzyme. So the transcripting enzyme that are found in the case of prokaryotic organisms is DNA dependent RNA polymerase. So from the DNA, RNA has to be formed. So RNA polymerase is required. But here, since the template is the DNA, so we say DNA dependent RNA polymerase, meaning this is the RNA polymerase which will make the RNA from the DNA. Clear? Like for this thing, transcription from DNA, RNA is made, right?
Yeah.
For this process, RNA polymerase is required because we are making RNA.
Yeah.
But where is that polymerase going to act? So it's going to act on the DNA.
Yeah.
So that is why DNA dependent RNA polymerase. Clear?
Okay.
So this enzyme, it can transcribe all types of RNA. So it can make every single RNA, whether it is tRNA, mRNA, or any kind of RNA. This particular enzyme, that is DNA dependent RNA polymerase, it can do the transcription of several types of RNA. Now, the process of transcription in the case of eukaryotes and in the case of prokaryotes, it's divided into three major steps: initiation, elongation, and then termination. So when we talk about initiation, so first of all, you have to remember this point, that like the RNA polymerase has two factors that are associated with it. It has sigma, which is called as the initiation factor, which will initiate the process of the transcription. And then it also has the rho factor, which is called as the termination factor. It will terminate the process or end the process of transcription. Clear? So during the initiation, the RNA polymerase, it binds to the promoter and it starts the transcription process, meaning that sigma factor of the RNA polymerase, that is going to bind with the promoter area of the DNA, and then it will start the process of transcription. Now the RNA polymerases, they are going to use nucleoside triphosphates as their substrates, meaning they will act on these nucleoside triphosphates that are present in the DNA, and it will start to make the RNA in a template dependent fashion, and it will follow the rule of complementarity. Complementarity rule is here G pairs up with C and A pairs up with U. Why U?
Because it's RNA.
Yeah, because it is RNA and RNA does not have T, right? So this rule of complementarity has to be followed while the RNA has been synthesized. Now how? First of all, the helix of the DNA, it opens, and then slowly, part by part, the whole entire DNA gets transcribed. The transcription of the whole entire DNA takes place. Now, after that, there is the termination. So once the terminator region of that DNA is
Voice
reached,
when.
you know,
Yes, ma'am.
So as soon as the terminator region of the DNA is reached, now the rho factor will come and it will cause the termination, and the RNA will fall off along with the RNA polymerase. And then the transcription has been completed. The RNA that is formed at the end of transcription, it is the mRNA. And this is very important that in the case of the prokaryotes, the RNA that is formed, meaning the mRNA that is formed, it requires no modification. The mRNA that is made, it will be used as it is. No modification, nothing of that sort is needed. Clear?
Ma'am, one thing, can you just explain the two last stages again?
See, look at this picture, you will understand it better. Firstly, what happens is that the sigma factor comes, right, and binds with the RNA polymerase and makes it active. Now it will bind over here and cause the transcription to begin. Now this RNA polymerase, it will move throughout the entire length of the DNA, thereby opening the DNA, transcribing that area, opening the helix, transcribing that area, that way. And it will move entirely throughout the length of the DNA. What does it do? For example, if this strand is open, so it will make complementary base pairs to this strand, like that. And then this will become the mRNA. Clear?
Which will become the mRNA? The strand which is open.
The strand which is, no, the strand against that strand. The DNA is not utilizing. Yeah, it's not being utilized. Against this means complementary to this, another strand will be made. This is mRNA. Clear? And then the rho factor would come. Huh?
Ma'am, it like automatically comes?
Which thing?
mRNA, it comes.
No, it is made, na. For example, DNA sequence has A, T, G, C, A, G, C, like that. So against it, U, then A, then C, then G, then U, then C, then G. This will be the sequence of the mRNA, complementary to the DNA. Got it? This is the DNA strand and this is the mRNA strand. It's being made. Got it?
Yep.
Then the rho factor will come and it will cause the termination. So the RNA polymerase, it is an enzyme, so it will not get used up. It will be as it is. It will come out, and so does the mRNA. This is the mRNA. This is the formed mRNA. Now, once this mRNA comes out, it requires no modification, nothing of that sort. Nothing of fancy and makeover, nothing. It's done. Now, this mRNA will be utilized to make the proteins. Is this clear?
Yes, ma'am.
Now, there is this question that in the case of bacteria, the process of transcription and translation, both of them, takes place in the cytosol or the cytoplasm. Why? Why not a defined or confined space?
Because cytoplasm is like packed closely, that's why like it has a cover so that it won't go out or something like that. Is it?
Because since it is a prokaryote, so it will not have
Your
any...
voice is like
Since it is a prokaryote, so it will not have any membrane, right? It doesn't have a nucleus. It just has the cytoplasm only. So that is why both of these processes takes place in the cytoplasm.
Can you repeat the answer on your voice? Like,
see, my answer is that transcription and translation, both of them will be taking place in the cytoplasmic area only, because there is no compartmentalization in the cell of the prokaryote. Meaning there is no membrane bound structures, right? There is no nucleus.
Yeah, that's what they're packed together. That's why it's taking place over there.
Also,
Okay.
many a times what happens, the process of translation or the protein synthesis, it starts much before the mRNA is fully transcribed. So for example, if this is the DNA, and from the DNA, mRNA is coming out. Now, it's not fully out, but this part will start undergoing the translation, meaning protein synthesis. Because all of it happens in one area only.
Yeah. Okay, so my answer wasn't exactly like wrong. Kind of correct.
Now moving on to the process of transcription in the eukaryotes. So the process of transcription in the case of eukaryotes, it takes place in the nucleus because now they have a membrane bound structure separated from the cytoplasm. So it will be taking place in the nucleus. But in the case of eukaryotes, it's very complex. Now there are RNA polymerase in the nucleus as well as in the, yes.
Just go a little bit up. Once again. No, the process of transcription. Okay. Yes, ma'am.
Hmm. So there are RNA polymerases. Like see, in the eukaryotes, there are cell organelles, and each of them have their own RNA polymerase, which makes this process of transcription very complicated. So three RNA polymerases, they are present in the nucleus. Okay, three of the RNA polymerases, they are present in the nucleus. What all are they? So RNA polymerase 1, this will be doing the transcription for 28S RNA, 18S RNA, and 5.8S RNA. These are the different types of RNA. So they will be transcribing these RNA. The RNA polymerase 3, it will be doing the transcription for tRNA, 5S rRNA, and SN RNA, that is small nuclear RNA. And lastly, the RNA polymerase 2, it will be doing the transcription of the hnRNA, that is heterogeneous nuclear RNA. This is the precursor of mRNA, right? So you see here that there are multiple RNA polymerases, each of which have their own RNA to transcribe. Got it?
And your voice is like going and then it is the thing which you're holding.
Is it fine?
Yeah.
So I was saying that since there are multiple RNA polymerases present, each of them have their own particular RNA to transcribe. So that is why they will be doing the different transcription, and that is why it is complex in eukaryotes.
So they do different at like different stages. Like not
No,
all at
all of it happens during transcription only, but it's just that all of these RNA polymerases, they come into play. Depending upon the need, obviously. If there is the requirement of tRNA in the cell, so the tRNA will be transcribed. And why would the other RNA be transcribed? Depending upon the need of the cell, the RNAs, they get transcribed. Clear?
Yes, ma'am.
Now, after the transcription is over in the case of eukaryote, like the basic step is that only. Initiation, elongation, and termination. After the transcription has been completed, the hnRNA that is formed, because we know that first the hnRNA will be formed and then only the mRNA will be formed, right? So these hnRNA that are formed, they contain special, or you can say extra sequences that are called as exons and introns. Out of these, the introns are useless. They do not have to be kept in the transcribed mRNA. So for the removal of the introns, the splicing process happens. Splicing, it is the process.
What is exons and introns?
See, for example, if this is the mRNA sequence, so in the sequence only there are certain sequences that are called as exons and there are certain sequences that are called as introns. Out of these sequences, the introns, they are useless. They do not have to be there in the mRNA. It makes the mRNA sort of, less efficient. So they have to be removed. Exons are useful. They are the coding or the functional sequences. So now the process of splicing has to be done. Now this is the modification of the mRNA that is taking place. Right? Mean not mRNA, you can say hnRNA.
Okay.
Right. So after the HN,
then mRNA, right?
huh?
RNA and then mRNA.
Yes.
Okay.
Yeah. Because hnRNA is the precursor.
Yeah.
So first splicing you've understood?
Yeah, it is. They just remove the introns.
After the removal of introns, the exons are joined together. For example, if this is the sequence, exons are there, introns are there, exons, introns. So if you remove this, the exons have to be joined together. Now, two additional processes are required that are called as capping and tailing. Now, in the case of capping, what happens is that we add, not we, the cell adds methyl guanosine triphosphate. Okay, MgTPPP. Right? They add the methyl guanosine triphosphate as the 5' end of the HNRNA. So it gets methylated. During the capping modification, the HNRNA, it gets methylated. Okay. Now this happens at the 5' end. Now at the 3' end, the tailing happens, in which the adenylate, like polyadenine, like a lot of adenine, such as 200 to 300 adenine residues are added at the 3' end. So at 5' end, methyl guanosine triphosphate will be added. At 3' end, the adenylate residues will be added. Clear?
Okay.
And now, after the splicing is done, after the capping is done, after the tailing is done, now the HNRNA is fully processed and now we have mRNA. Now, since this process was happening in the nucleus, now it has to get out of the nucleus and come into for the translation process, okay, which is protein synthesis. And the translation process will be taking place in the cytoplasm. Clear? Any doubts?
No.
The presence of introns is reminiscent of antiquity. And it's like a very significant thing in terms of the eukaryotes. And the process of splicing, it represents the dominance of RNA world. It means that this suggests that it was the RNA that was initially the genetic material, and it proves that. Clear? Splicing process. Why? As the RNA decides to remove the introns and keep the exons. So it's like showing power, showing dominance. Okay, I can do so. DNA doesn't have that choice. DNA doesn't need it also. Any doubts up until now?
DNA doesn't need it, right?
Now, coming on to the genetic codon. Now see, yes.
You put it over here only. The thing.
My hands are pain. Okay. Is it okay
You
now?
can take a tape and just
Wait,
put it.
I'll just put it on my ear like that. Is it fine now?
Yes, ma'am. Yes, ma'am.
So, as I was saying, that is it okay now?
Yeah.
Okay. So, every genetic code, meaning if you talk about DNA, okay, if I write the code for DNA, it is always read in a triplet manner, like AUG, GUG, CUG, like that. Always in triplet, never in any way or. So,
Yeah.
this is a genetic code. So, we will now study the features of the genetic code. Now, the genetic code, always remember that it is in the form of a triplet, meaning 3 nitrogenous bases read together. And when you read the triplet, it is always read without any punctuation, meaning you can't stop between the A and U or U and G or anything of that sort. Okay. After that, the genetic code is degenerate. Degeneracy means, see, for example, you would think that every genetic code is unique, but that's not the case. There are some genetic codes that are, coded by more than one codon. Like some amino acids, they are coded by more than one codon. So, for example, methionine, it is an amino acid. It is coded by the codon AUG, and this AUG is also the initiator codon. So it is coding for 2 things. So it is degenerate, it's not specific. Yeah. Now, It is a universal code, meaning the genetic codon is a universal code, meaning that if we write UUU, so that is the code for phenylalanine amino acid, and starting from the bacteria up until the human beings, phenylalanine is coded by UUU. Getting it?
Yeah.
Then comes the initiator or start codon. So this AUG, it is going to be coding for the methionine as well as it is the initiator codon. So whether we talk about replication, whether we talk about this thing, transcription or anything of that sort, the process cannot begin if it is not started by the start codon. So that is the significance of the initiator codon or the start. It will start any kind of molecular process. Without this, it won't happen. Is it clear? Hmm? Huh?
Could you repeat?
I was saying that this initiator codon, no matter whichever process it is, that process gets started because of the presence of initiator codon. It will initiate any kind of molecular activity. Without this, no molecular activity will be started. Now, stop signals or terminator codons. Now, there are certain sets of codons that we call as the stop codons. What are they? UAA, which is called as ocre or whatever you want to say. UAG, which is amber, and UGA is opal. This is like the names of the people that have found them. So, these are those codons that do not code for any amino acid, and we call them as nonsense codons. But wherever they will be present, they will stop the process. So for example, see this. If this is the sequence of DNA that is undergoing transcription, AUG is there, it will start the process. And then UUU will happen and all that. Then as soon as the UGA will be reached, the process will be stopped. Why? Because it is a stop codon. Now no more transcription or translation will happen. Okay? So these are certain properties of this thing. Features of the genetic code. Any doubts? Then comes mutation. Now we have already, I think I have taught you mutation. Have I?
Yeah, we teaching. Yeah, last chapter.
So mutation, so shall I skip it or shall I do a quick revision?
Quick revision. I mean, I already finished reading. Okay, no problem. You can still do it.
See, mutation in general, this should be like on the tip of your tongue that it is the change in the genetic sequence of an organism, right? It is the altering of the DNA, and that it is sudden, first of all, it is directionless, right? And it is spontaneous. Now, in the case of mutation, the genes can either be lost or they can be gained. If they are being lost, it's a deletion mutation. If they are being gained, it is a addition mutation, right? Now, further, the mutation can be point mutation or frame shift mutation. Now, point mutation is when only a single base pair is being changed, one, right? Like for example, like a change in the single base pair in the gene for beta globin chain, right? Sickle cell anemia, you remember? The glutamate changes from the glutamate, it changes to the valine, right? Because of which the RBCs, they get a sickle shape. Then comes the frame shift mutation. So when either insertion is happening or deletion is happening in the multiples of 3 or more. And that results into the whole entire change in the reading of the frame. Like from triplet, it will go to doublet and all of that. So that is called as frame shift mutation. It will shift the reading frame of our codon. Clear?
Yes, ma'am.
Any doubts?
No, ma'am.
Now, frame shift mutation can be because of insertion or deletion. Then comes the tRNA. Now, the tRNA, it is also called as the adapter molecule, and 15% of the total cellular RNA is the tRNA. That the existence of tRNA, it was again noticed by Francis Crick. He was the one who said that, okay, tRNA exists. Apart from that, tRNA has multiple names such as soluble RNA, right? The tRNA is responsible for reading the code, right? And it binds. It also transfers the amino acid. It brings the amino acid or codons during the process of translation, that is protein formation. Now, there are 60 different types of tRNA that are present. And now when we talk about the structure of the tRNA, so it is inverted L type and it is clover leaf shape. So the tRNA can be a variety of, like their structure has been defined in such a way. Actual shape or actual structure is the inverted L shaped structure, and the clover leaf shaped structure, it is the secondary structure of the tRNA. Now, where is the picture?
Is that thing
See
which you
this.
prepared?
Huh?
Clover leaf shape is that thing
This
which you
one.
wrote. Oh, that.
It is the clover leaf shape, roughly. You won't find exact shape. And this is the inverted L shape. Okay. Now, if you see in this picture, there are loops, right? You have T loop, D loop, anticodon, and anticodon loop is there, right? So each of these loops have a particular function to perform during the process of translation. Okay. Now we are going to see how. First of all, the amino-acyl synthetase binding loop or the DHU loop, dihydroxyuridine loop. This is the first loop from 5' end. 5' is here, right? This is the 5' end. So the first loop is the DHU. This is D loop, or you can call it as DHU, dihydroxyuridine loop. It is the first loop from the 5' end of the tRNA. Okay. And then there is the ribosomal binding loop or the T-chi-C loop. This one, T loop, or you can call it as T-chi-C loop. Okay. This is the loop that binds the ribosome. Then after that, there is the anticodon loop, the one at the bottom. It has bases. Now, this part or this loop of the tRNA, what does it do is that it actually codes for the proteins. Like whatever the protein codon has to be made, it is actually coded by this particular anticodon loop. Is it clear? Did you get it?
It has to bind to the amino acid at the end.
It binds to the amino acid because like 20 amino acids come together to make one protein. So the binding of amino acid is done by this loop. Got it?
Yes, ma'am.
Any doubts?
No, ma'am.
Okay. Now, after that, let us look at, now see, this inverted L shape, it's actually the 3D structure, whereas this one is the 2D structure. For the sake of understanding, we see the 2D structure, but the real structure is the 3D. Okay? Both of them are actually the real structure. It's just that for the sake of understanding, we learn better when things are flat, isn't it?
Yeah.
Now let us look at the process of translation. Now translation is when the amino acids are polymerized in order to form a polypeptide. Polypeptide means, what is the meaning of polypeptide?
Many peptides together.
So what is it? Many
Polypeptide.
peptides. What is a polypeptide? The protein.
Yeah. Oh, like that. Okay.
So it is like the translation, you can call it as translation or you can call it as protein synthesis. Now it takes place in the ribosome of and you know that the eukaryotic ribosome and the prokaryotic ribosome, it's very
We
different.
did it, ma'am. We did it. Is it from the mRNA to proteins? Now it is going from mRNA to proteins, right? Translation.
Yes.
Okay. Okay.
Because ultimately from the mRNA, the protein has to be formed. This will complete the expression of the gene when the protein will be formed. Only the mRNA is useless. Why is the mRNA formed? It is formed so as to form the protein. That's its main function. Now, the ribosomes of the eukaryotes, they are of ATS type and it has two subunits. You know that it is made up of 60S and 40S. 60S being the largest subunit and 40S being the smaller subunit. Ribosomes, they have different sites that are present in them. Okay, then they have T site, which is the peptidyl site. They have the A site, which is the aminoacyl site. So in the ribosome, there are sites present. Okay, that will be like when, so for example, this is ribosome, this is mRNA. So this is mRNA. When the ribosome will come and sit on the mRNA, the sequence that it is sitting near to, it will synthesize the protein against that. Then it will move against that. Then it will move against that.
Can you tell that again?
Huh?
Thing again. Can you tell that again?
I was saying that the ribosome, it has certain sequences that are present in it or binding sites that are present in it. Okay. So according to the binding sites, it will bind to the mRNA and make the protein. So if I'm saying that this is a ribosome and this is the mRNA. So the ribosome will come, sit on the mRNA and the part that it is sitting on, it will make proteins against it. Then move forward again.
Oh, okay. I understood. That. So, but the ribosome will be of 60 and 40S like that.
Those are subunits.
Oh.
60S and 40S are, are the subunits of the ribosome of eukaryotes.
Yeah, so that will come and it will bind for that, right? Okay.
And whenever the smaller, like as soon as the smaller subunit of the ribosome, when that will be binding with the mRNA, then and there only the process of translation will begin. Then and there. Clear? As soon as
Ma'am, the whole mRNA won't be translated, right? Only
course
the
it will be.
parts where the ribosome comes.
So it will go to the whole entire mRNA. No?
It will go like this.
It
Oh.
will actually go like this.
Oh, okay. So it
mRNA
complete like the up and the down is taken. That's why. Okay.
throughout. Okay.
Mm.
Now the amino acids, they are joined together by peptide bonds. Okay, peptide bond is the bond that is present in the proteins. And those bonds only are responsible for making the protein. Clear?
Yeah.
Now the formation of this peptide bond, it requires a lot of energy, that is in the form of ATP, right? Obviously. Now let us see the steps of translation. Now firstly, the amino acids, they have to be active. And when does the amino acid becomes active, or when does the activation of the amino acid takes place? It takes place when the amino acid binds with the ATP molecule. And as soon as the amino acid binds with the ATP molecule, then the process of translation is like, okay, then the amino acids have become activated. After that, simultaneously, the charging of the tRNA also has to take place. Meaning, when that activated amino acid which we just saw, when they will come and get linked with that tRNA, the normal tRNA, right? And then we say that the tRNA has been charged. Since the activated amino acids are being added here, we call it as amino acylation of the tRNA. Getting it?
Can you just write in bracket amino acid plus ATP?
No, amino acid plus tRNA. For charging of tRNA, it is amino acids that come with tRNA. And here in the activation, here amino acid plus ATP.
ATP. Oh, okay.
Then comes the formation of the polypeptide chain. Now, after this process is done, now the ribosome will be acting like a catalyst. It acts like a catalyst, and what will it do?
Make
Basically,
the process fast.
make the process faster, or actually cause the process to be happening. If it will not be there, how the process will happen at all,
Yeah,
right?
it won't be. Yeah.
So the ribosome, apart from obviously being the place where the protein synthesis is taking place, right? So at that point of time or at that place, it will act like a catalyst as well. Now in the bacteria, 23S RNA that is there, okay, it is a catalytic RNA. And a catalytic RNA means it is the RNA that acts like a enzyme, and we call it as ribozyme. Clear? Are you getting it?
Can you repeat this
No.
one?
See, first of all, you understand this much, that the ribosome, it acts like a catalyst. Clear?
Yeah.
Now, there are certain RNA in the bacteria that have this catalytic property as well, and we call them as the ribozyme. The catalytic RNA which has the enzymatic or catalytic property, we call them as the ribozyme. This is just an additional information not related to the first line.
I still didn't understand it. I understood ribosome acts as a catalyst. Yeah. Then after that?
See, this, like in the case of bacterial, meaning in the case of prokaryotes, there is this rRNA, ribosomal RNA, which have this property of acting like the enzyme,
Okay.
which is called as ribozyme. RNA enzyme is called as ribozyme. Usually enzymes are proteins.
Okay. Understood. Okay.
Now, a translational unit in the mRNA is the sequence of the RNA that is flanked. Flank means to, like, for example, if this is mRNA here and here, this is flanking. Many things are put like that at the two ends, right? So that is flanking. So the translational unit, meaning the part of the RNA that has to undergo translation, it is the sequence of the RNA that gets flanked by the start codon at the beginning and at the end stop codon.
Why does it go in that thing?
Because it has to get started and stop.
No, like
If the stop codon will not be there, how will the process stop? The process, see, for example, if the mRNA is that long, but the protein requirement is this much. So here and here, there will be the start codon over here, there will be the stop codon over here. So once the translation is done till here, the process of translation will stop. The whole entire mRNA will not undergo it. Or let's just say the entire length of the mRNA, this is how it's regulated. There's no Y in it. This is how it's like it is. Right? Start codon is there obviously to start the process. Stop codon is there to stop the process. Yeah. And this translational unit, it will be coding for that protein that we require, the cell requires. Now, an mRNA, it has some additional sequences that do not get translated. Like throughout the mRNA sequence, there are some that are not going to get translated, and we call them as the untranslated region, UTRs. And they are present at the 5' end also, like before the start codon, and 3' end also after the stop codon. So those areas of the mRNA, they are never going to get translated. The place where the start codon is present, before that, whatever sequence is present will not get translated. The place where the stop codon is present, after that, whatever sequence is there will not get translated. And these untranslated regions, they're very important as far as the efficient translation is concerned. Like they make the process of translation efficient. Clear?
I could not hear that. Sorry, what did you say?
What are you thinking?
No, like they start and they stop, and there are codons before them and after them. So they are not
Because
translated.
what I said, that the process,
Thing
that
only
start
this much is
codon,
required.
the
That's why.
start codon will start the process. So like wherever it is present, from there only the process will start. Anything that is before it will be not translated because
So
the
that would
start
be like
codon is not
untouched,
present.
just left like. Okay.
Not untouched, it will not get translated only.
Okay.
Clear?
Yeah.
Now looking at the steps of the translation. So there is chain initiation, elongation, and termination. Now, during the initiation, the ribosome will come and it will bind to the mRNA where the start codon is present at that region. Now, the initiated tRNA which is there, this coming of the ribosome binding to this area, it will be recognized by the initiated tRNA. Now, the ribosome will start the elongation part, meaning, okay, now it has come. It is attached to the AUG, that is the start codon, and it will begin the process of translation. Now, during elongation, the amino acid and the tRNA, they will bind to the codon in the mRNA and start forming base pairs with them. Again, A with T, G with C, like that. The ribosome will start moving from codon to codon, codon to codon throughout the entire length of the mRNA. And in this way, the amino acids, they are getting added one by one to the mRNA and they are being translated into the proteins. At the end, the release factor comes to the stop codon and the translation gets completed. Okay. All right. So now the translation process gets terminated. So now at the end of the translation, what we are left with is a proper complete polypeptide, which is the protein. Is it clear?
Yeah.
Now see, there is this question that says write the sequence of the complementary strand in the 3' to 5' direction. That you can do. Complementary sequence.
The AUG, that thing.
Like this is the sequence that has been given to you and you have to find out the complementary sequence. So this you can do, na?
Oh, so the next strand, right?
Complementary sequence, not the next strand. Like this is 5' to 3'. So you have to find 3' to 5'. Can you do that? Do it.
So the given strand is CG. I just have to shift it, like, right from.
You have to make complementary basis. You have to make to the complement, like C complementary with G. So G is the there. Then G with C.
Oh, okay.
And do this part also. Write the sequence of the transcribed mRNA of the same sequence.
Okay. Is it the correct one? GCATAGCTGCCAA
Wait.
GCT.
You check. GCATGCCAATGCT.
Wait. Ma'am, after see, I wrote GCATAGCTGCCAA.
Wait. What will be the complementary of C over here? Say you've written it correctly. Say what will be the complement. See,
One,
listen.
two, three, four,
Speak
five,
it
six.
slowly. Speak it slowly.
G.
Hmm?
G.
Then?
Then C.
Then?
T.
Then?
G.
Okay, then?
C.
G, C, okay.
Then again C.
I am saying you check. Okay? Getting very hodgepodge.
I'm getting
The
C.
first C, listen, now you look it here. Okay, finals.
Okay.
G, C,
Okay.
A,
Okay.
T,
Okay.
A.
Okay.
Right?
Okay.
G, C,
Mm,
T,
mm,
A.
mm.
This is where you are
Then,
doing wrong.
yeah. I don't understand. Wait. Did I write for? There was a T there? Oh, I copied the script wrong. Sorry. Sorry. Wait.
So this is
T,
so this is the
C,
sequence. Now
G.
what have you found out? Like once you have done the complementary sequence, it is the sequence of what?
Um, the next sequence, right?
It's the complementary sequence. Like one is in the 5' to 3' direction.
Yeah,
The
one is
other
in
is
the
3 to 5.
Yeah.
So you found out the 3 to 5 direction.
3 to 5.
Now you find out the mRNA sequence.
Oh, so replace the T with the U like that.
Of which strand?
Same thing.
5 to
Of
3 or 3 to 5?
the 5 to 3. 5 to 3. Because that will be the same as the mRNA, right? Just the T has to be replaced with U.
Yeah, exactly.
Okay.
Clear?
Yes, ma'am.
Any doubts?
No, ma'am. Okay. CGUAUCGAUCGGUUACGA. Nice. Finished.
Got it? These kind of questions can come. So you will be able to solve. Just try to remember the words, what they are saying. Right? Whether they are saying complementary and complementary of what? If it is from 5' to 3' or 3' to 5', or they are talking about the complementary sequence of the mRNA.
They will give the thing, right? CGT.
Obviously, they'll give, like, how will you do it? They'll not.
I
Okay,
don't know.
now
But
wait. I have few questions that you need to make. Can you see the screen? Now try to attempt these questions. A major portion of your chapter has been completed. We are only left with the Human Genome Project. So I am not going to do that
Voice
today.
is like, um,
I'm saying a major portion of the chapter has been completed, right? And the rest of the things, I think only the Human Genome Project is left and DNA fingerprinting. So I will do that in the next class. Till then, you solve these questions, these MCQs. You just write the answers.
um,
Yes.
should I tell the answers?
Yes, please. If you have any doubts, tell me that also.
Okay, so the first one, is it phosphosidyl bonds or is it peptide
Which
bond?
bond?
Second one or the third one? I
No.
think it's the
Which
second
one?
one.
Read it.
Phosphos Phosphodiester bonds.
Phosphodiester bond. Okay.
Ester
First one
bonds.
or second one? What are they talking about?
The linkage. Nucleotides are linked together by DNA strands. DNA
Hmm.
strands. Phosphodiester only. Peptide bonds are not.
Obviously.
Yeah.
Yes, phosphodiester. Next.
Yeah. Then it is a phosphate group. Nucleoside differs from nucleotide. Yeah. Then it is pentosis.
Pentose is meaning how many carbons do they have?
5.
Very good. Okay.
Um,
What is the answer? The fact that a purine base always pairs through hydrogen bond with the pyrimidine base. This leads to what
Wait,
does
wait.
this purine base pairing with pyrimidine confers to the DNA?
Okay, throughout the DNA only. C, option C.
Yes. The net electric charge on DNA and histone.
Ma'am, histones are positive.
Hmm. This is a very easy question.
Negative and positive respectively.
Yeah, obviously the histones, they are positive and DNA has to
And
wrap
DNA
around
has
the
to be
histone.
negative. Yeah,
Yeah,
exactly.
then only it will wrap around. Okay, next. Upstream means towards the place where it will begin. Downstream is like towards the end.
Oh, 5 upstream and end and 3 downstream end respectively of the transcription unit.
Very good. Next.
Sickle cell and malaria. Okay. All of the above. What is acquiring malaria? See,
Getting
two
malaria.
options are correct. See, it cannot be treated with iron supplements. It is a molecular disease. Yeah. I don't know what acquiring with malaria is. So all of the above. I don't
It confers resistance. Okay. Have you been taught human health and diseases in school?
What?
Have you been taught human health and diseases in school?
Uh-huh.
See, what happens is that this is a very nice question. It's saying that which of the statements it is appropriate for sickle cell anemia. You know that in sickle cell anemia, the RBCs, they go from biconcave shape to the sickle shape, right?
Yeah.
So first two, obviously they are correct. The third one
Yeah.
says it confers. Confers means provides resistance. Resistance means the ability to resist something, meaning not let that happen. To acquiring malaria. Acquiring means to get something. And malaria, see, malarial parasites, they feed on the RBC. And if the RBCs have a tiny, tiny space, malarial parasite cannot feed there. I mean, not actually feed, reproduce. So that is all of the above.
So it
Did you get
provides
it?
resistance in malaria.
It provides resistance, like meaning those people that have sickle cell anemia, they will not have malaria because their RBCs are not enough for the malarial parasite to reproduce.
Wow. Okay. Okay.
Next.
Is an initiation program. Wait. I'm not giving a fair answer. Wait.
I see that you make tough questions very easily, and the questions that are very easy, you get stuck on.
Yeah.
I don't know. This is your classic Rida move.
I know, right? See. Okay.
What is so confusing in this
It is
question? It's
all
so
of the
straight.
above. All
Yeah.
of the
All of the obviously AUG is a starter codon. It codes for methionine also, and it initiates the process anywhere. And since you know that the codons, they are universal, so this will be coding as a start codon, initiator codon for all of the organisms, whether it is a prokaryote or a eukaryote.
No. See,
Simple.
I knew the first two, yeah, but then I was like wondering if it's for prokaryotes and eukaryotes, and I was thinking if eukaryotes have another set
Always
because I
remember that they are the codons. They are what, universal.
yeah.
So whatever, like whether it is like
Yeah, no, because my brain,
phenylalanine in
yeah,
prokaryotes
no,
also.
no, my brain decided to think that, eukaryotes, how can there be U in it? And then, yeah, it went into another thing.
Okay, next. The first genetic material could be
Uh, RNA.
hmm. With regard to mature mRNA in eukaryotes.
Oh, exons appear, but introns do not appear in the mature RNA.
Yes, why?
Because the introns, they
Use
go
the
away.
right term.
Okay.
Go away,
I mean,
come
it,
another day.
no, introns, they they just get removed.
No, you have to use the right. No,
Disintegrate.
no.
This,
What happens to the introns?
they removal of
So
intron.
what is it called? The removal of introns is called as
Something I don't remember.
splicing.
Oh, okay, yeah, slicing. Yeah, I learned it
This
today. See, that's why.
this next question is not relevant for now.
Okay.
Who amongst the following scientists has no contribution in the development of double helical model for DNA?
Wait. See, I know New Zealand.
Now you have to tell me the contribution of all of them.
Okay, Rosalind Franklin, she found out the X-ray of the double helical thingy. Oh, wait. So it's Messelson and Stahill, because Erwin Chargaff also was there. Morris Wilkins also was there. Erwin
What did
Chargaff,
Morris Wilkins do?
he um the pairing of the base pairing rules and all that. He did that. So
Wilkins and Franklin, both of them, they gave the
yeah,
X-ray
they did.
crystallography data of the DNA,
Yeah,
based off of which Francis Crick deduced the double helical structure of the DNA, right?
yeah,
Erwin Chargaff told us about the complementarity rule, right? The Chargaff's rule. And
yeah.
what did Matthew Michelson and Franklin Stoll do?
I don't remember. I just
Michelson and Stoll,
remember
they did an experiment. Which
of
experiment?
the rat thingy. I think.
Right? He transforming
Yeah.
principle.
Transforming principle. Yeah, the rat.
Huh.
I mean, as
Okay.
this
So
thing.
your
Yeah.
answer is D and it's correct. Okay. DNA is a polymer of nucleotides which are linked to each other by 3 to 5 prime phosphodiester bond. To prevent polymerization of nucleotides, which of the following modifications would you choose? Polymerization.
Both B and C. Can't replace it by remedies.
No, not C. Why? Because it is already a DNA, so it doesn't have that 2' OH. B would be correct.
No, B and C. No, you
Why B
remove
and C?
or replace the two OH group with some other group in deoxyribose.
So deoxyribose itself means that the 2'
Itself is
OH
there,
is
yeah.
not there, na?
Yeah, but
So why, if it is not there, so how will you replace it or remove? If it's already not there, you can go for the 3' OH so that they don't bind with another groups. The OH and the H can form a bond and become H2O,
yeah.
right? But the 2' OH that is not there in the first place, so how will you remove or replace it? It's deoxyribose sugar. Had it been ribose sugar, then you could have said the same. But it is deoxyribose sugar, na? So they don't have it in the first place. Discontinuous synthesis. Wait. Have I taught you DNA replication, Okazaki fragments and everything?
Yeah.
Discontinuous synthesis of DNA, it occurs in one strand. Why?
Wait, you did not teach Okazaki fragments.
That is what I'm saying. I don't
Yeah.
remember teaching it to you. I have not taught replication.
Yeah, but like
Yeah.
I know it's some sort
You
of.
know it, but I have not taught.
Yeah.
Okay. I have one more topic to do with you. Huh. So tell me if you can answer. Tell. If you don't, I will not blame you.
B.
Correct. Because if
I
it is
don't know.
happening in this direction,
It cannot like
that is why small fragments are there.
yeah.
Then these fragments only, they are called as Okazaki fragments. Later on, they are joined together by DNA ligase.
Yeah, but then for 5 to 3 prime, it is going in a straight thing only because like
Because the
it
whole
can.
entire process of polymerization or the replication, it takes place from 5 to 3.
Yes.
Which of the following steps in transcription is catalyzed by RNA polymerase?
Wait. Which of the following two steps in oh sorry. All of these, yeah. Finish. Yeah. Done.
Really?
Yeah, all of the above.
No. How? They are saying which step of the transcription is catalyzed by the RNA. So it's elongation, na? The RNA polymerase is doing the this thing addition of the base pairs in the elongation phase. In the termination phase, the rho factor comes and the RNA polymerase go out.
Oh, yeah. Okay, correct. Yeah.
In the initiation, there is sigma factor, but it is
Yeah.
through and through going here. Got it?
Yeah.
Okay. Control of gene expression. This is lactoperon is also left. This is from lactoperon. So this, like, now the rest of the questions that are there, we have to do the topics, the remaining topics, and then we'll complete these questions. Up until now, do you have any doubts?
No.
All right. So we'll be keeping it till here only. The notes. I saw that you were having trouble in the fact that, you had to write here.
Yeah.
Hmm.
No, because our teacher, she finished evolution. Okay, she finished this long back. She finished evolution and now she's doing reproduction in flowering plants.
Okay. So these notes, I will send it to you, but please don't get them printed out. Write it
Yeah,
whenever
I'll
you get
write
time.
them. I've been writing them. Ma'am,
All right.
the previous notes which they sent, no, the previous chapter, those notes are blurred.
They're blurry.
Yeah, they have
I'll
not
see
come.
to it. Okay, I'll see to it. I'll send it to you. Okay. So, all right, we'll be keeping it till here only for today's class, and we'll continue in the next class.
1 second. Can you just pray that I get the head girl position? Like tomorrow they're gonna give out the candidates' names.
Like tomorrow is the result day.
Yeah, result day. Like
Will you let me
not
know?
like select like will like 9 people have given interview. So out of those 9 people, teachers will select now.
Shortlisted people.
Campaigning. Yeah.
Inshallah. Okay. I will pray for you. All right.
Jazakallah.
Okay. All right, dear. I'll see you in the next class.
Bye, ma'am. Take care.